My name is Taylor Barad, and this video is on my Bio 205's cardiovascular physiology project on the effect of ice on heart rate and reaction time. The principal investigators in this experiment were myself, Taylor Barad, and my lab partners Laura Goldman and Sydney Reed. Our TA is Elyssa Hopkins. Some background information that I'd like to go over before I switch to the PowerPoint is that we ran this experiment 2 times. The first time in Lab 21 was for us to run the experiment, get comfortable with the equipment, and to find where there were some experimental design errors. The main error that we found, is that we immersed the subject's hands in ice for 1 minute, and then we recorded their heart rate and reaction time. What we found was that the hands began to thaw between the heart rate and the reaction time measurements. So a big change that we made from Lab 21 to Lab 22 was that we re-immersed our subject's hands into ice for 1 minute after heart rate measurements and before reaction time measurements. So now I'm going to go switch to the PowerPoint and present our poster, and I will talk over that. Our experiment is on the effect of ice on heart rate and reaction time. The purpose of our experiment is to understand how a person's heart rate and reaction time will react after exposing one's hands to ice for 1 minute. Before we started our experiment, we did some research to help formulate our hypothesis, and we found that when the external temperature of an environment is decreased, the heart rate should decrease as well. We also found that in freezing temperatures, a person's reaction time will increase, and this is because the nerve signals from the brain are slowed down, thus slowing down the muscles, which will, as you'll see in our method section, it'll slow down the time that it takes for the hand to click on the mouse, which will increase our reaction time. So the hypothesis of this experiment is that after a subject's hands are immersed in ice, their heart rate will decrease and their reaction time will increase. So here's our method section, and basically for our experiment, we measured the subject's heart rate and reaction time before and after their hands were exposed to ice for 1 minute. So the subject's heart rate was measured using an ECG, which was connected and measured through LabTutor, which was provided by Bio 205. The reaction time was measured using an online reaction time test, which calculated the average of 5 trials. So what this online reaction time test did was there was an orange screen, and the person would stare at the screen, and every time that a green screen popped up, the person was supposed to click the mouse. So the person did this 5 times, and the computer took the average. So after we took the heart rate and the reaction time before being exposed to ice, the subject was then exposed to ice for 1 minute. After 1 minute, we measured the subject's heart rate, and then we put their hands in ice again for 1 minute to ensure that the hands did not thaw, because this was a problem that we found in Lab 21, which I mentioned before. So after that 1 minute, the reaction time was then recorded, and the subject was allowed a break because we ran this experiment twice. Next, I will go over our results. Shown here is Figure 1A. Figure 1B, which is coming up next, both represent the results obtained by Trial 1. Here in Figure 1A, the blue bars represent the heart rate before exposure to ice, and the orange bars represent the heart rate measured after ice. Subject 1 was the control group, so it doesn't matter whether or not the heart rate increased or decreased because there were no changes made to their environment. Their hands were always at regular body temperature. Subjects 2 and 3 both experienced a decrease in heart rate after they were exposed to ice. Here in Figure 1B, we're showing the reaction time before and after the ice, Again, blue represents before ice and orange represents after ice, and subject 1 is the control group. Subject 2 had an increase in reaction time after being exposed to ice, while subject 3 had a decrease after being exposed to the ice. Now, for figure 2A and 2B, we're going to be talking about the results obtained by trial 2. So again, the blue bar represents before ice and the orange bar represents after ice, and subject 1 is still the control group. So in this figure, figure 2A, which is showing the heart rate before and after ice, the heart rate increased for subject 2 and decreased for subject 3. Here in figure 2B, the reaction time increased for subject 2, but it decreased for subject 3. Shown here are the t-tests that were made for the heart rate before and after the subject placed their hands in ice water during trial 1, and the t-test of the heart rate before and after the subject placed their hands in ice water during trial 2. On this slide, here are the t-tests of the reaction rate before and after the subjects placed their hands in ice water during trial 1, and again, the t-test of reaction time before and after the subject placed their hands in ice water during trial 2. I will go over the significance of these results in the next section. So now I'm going to talk about the conclusion of our experiment. Just as a reminder, in our experiment, subject 1 was the control that did not undergo exposure to ice. As seen in our results, in trial 1, there was an overall decrease in both reaction rate and heart rate. In trial 2, there was an overall decrease in reaction rate, but an increase in heart rate. In both trials, after doing the t-test, the t-stat was lower than the t-critical value. Thus, we cannot reject our null hypothesis. There are many reasons why we got the data that we got. However, it may be due to the fact that only the hands of the subject were exposed to the ice as opposed to the entire body or the face. The core and central body temperatures remain the same at a typical body temperature, so the heart rate may have been unaffected. We did some more research and found that similar studies have shown that low temperatures sometimes have been insignificant to heart rate. Because our heart rate was not affected and the core body temperature did not drop, the reaction time was also not affected because there was no reduced heart rate or nerve signaling. So therefore, our hands were able to function regularly. Some ideas that we have come up with for future experiments are ones that involve the entire body. For example, an experiment that should be done involves complete ice immersion of the entire body before measuring the heart rate and reaction time. This is because cardiac function, which is based on enzymes in the cells, depend greatly on body temperature. So we want to see how cold temperatures compare to optimal temperatures. Another thing that could be explored is the topic of how heart rate and reaction time react to an increase in temperatures, such as high temperatures, because the body pushes itself in a different way because the body needs to cool itself down in order to maintain homeostasis. Thus, we hypothesize that the heart rate may increase and the reaction time, again, would decrease. Now I'm just going to show you our works cited. Again, thank you for watching my presentation, and I want to just extend a thanks to my TA, Elyssa Hopkins, everybody in Lab 28, and Bio 205. Have a good day.